In this work, we have developed a workflow to address the structure elucidation problem of metabolites. Metabolites are the small molecules that are intermediate or end product of a metabolic reaction that naturally occur within the cell. This figure shows that metabolic state that exists in the cell, and it is called metabolic dark matter because most of the state are currently unknown. So looking at the figure, you can realize that how challenging is that to predict the structure of metabolites. Here we have developed an in silico workflow to predict the structure of an metabolites, and our methods are taking the best aspect of the force field based method, QM machine learning based methods, and the standard QM methods to predict the structure of a unknown metabolites very accurately. This workflow consists of a 6-step. First step is the generation of the conformation, and metabolites are very flexible molecules, so the large number of conformational space exists for a particular metabolites, and therefore the accuracy of a result, that means the prediction of an accurate structure, depends on the how much conformational space that you have considered for a particular metabolites. So in this workflow, we have generated the conformation using the macro model that is integrated in the Schrodinger. Macro model is very widely used conformation generation tool for small organic molecules. So using macro model, we have generated fairly large number of conformation of a particular metabolites, and then we filtered all the conformation that we have generated by the macro model, which is the force field based methods, and filtered it by QM based machine learning methods called ASE-ANI. ANI is very highly accurate machine learning model that can calculate the chemical properties of the molecule very accurately. The accuracy is similar to couple cluster method, but ANI is the billion times faster than the standard QM method. After filtering all the conformation by the ANI machine learning model, we clustered it to get the unique structure of the metabolites. We clustered using Schrodinger. We have chosen the centroid of each cluster to get the representative conformation of a particular cluster. After clustering, we got the structurally distinguished conformation of a metabolite, and then we optimized it by standard DFT optimization. After geometry optimization, we calculate the chemical shift of the nuclei. Here we have calculated the proton NMR chemical shift and C13 NMR chemical shift. Finally, we Boltzmann averaged with all the conformation and predict the structure of the metabolites. So as a test case, we have the results of O-succinyl L-hemoserine. This is a very flexible molecules, and we have generated a 500 conformation of this molecule, and then we filtered it by any machine learning model. So we ended up with 485 conformation of this molecule. Then clustered it, and we got 25 unique conformation of O-succinyl L-hemoserine, which undergoes standard DFT optimization and followed by the NMR chemical shift calculation of the proton as well as carbon NMR. Boltzmann averaged throughout all the conformation and predict the structure of this molecule. To validate our results and to see the goodness of the fit of our results, we compare our results with the experimental values available, and we have calculated the MAE, which is mean absolute error, which is the difference of the calculated value and the experimental value of NMR chemical shift of a particular nuclei, and we have seen that our calculated results are highly accurate. In conclusion, this workflow is very effective and computationally very efficient. In the case of O-succinyl L-hemoserine, which has very high conformational space, and we have considered 500 conformation for this molecule, using our workflow, we can calculate the NMR chemical shift and predict the structure of this molecule by less than a day. But if you want to go by a traditional methods, it may take more than 10 days to assign the structure of a molecule. This workflow considers the large conformational space of a metabolites. As I mentioned previously, that metabolites are very flexible, so accuracy of the results depends on that how much conformational space you consider. So this workflow allows you to consider large conformational space. Users are not restricted for a particular DFT methods. So user can choose any of the DFT method of their choice, any of the functional and basis set combination, and user also can calculate NMR chemical shift in the solvent. So solvent model is included in our workflow. Overall protocols are fairly automated by Python based scripting. This workflow has a potential to calculate the NMR chemical shift of an unknown molecules and predict the structure of an unknown very accurately. The whole protocol is designed in such a manner that it can build an in silico library for a fairly large number of molecules. This work has been published in Analytical Chemistry, a journal, and this work is funded by National Institute of Health. Please check out this paper to learn more about this research.